My name's Andrew, I'm from Glasgow. Evolved from basic clinical medicine, genomics, now really focusing on implementation. And I'm going to ask you guys to do something different because I tend to talk too much. I'm going to ask you to read the first set of slides. So we've moved from developing therapeutics, which we still do, to really trying to solve the problem, which is not scientific. We do great science. I've published enough papers in my life. Sometimes people read them. Have they made a difference? No. So the patients know what needs to be done. But we as a community, as a health system, aren't listening. To try and address this, we had a TEDx lab or a TEDx session in Glasgow earlier this year, really bringing in patients, policymakers, others, to really try to get to the nub of this problem and really starting to listen to what our patients really want. And they know what we want, but they get it more than we do.
Hi, I'm Leslie Stephen. I live in Edinburgh with my husband and 4 children. And over 5 years ago, I was diagnosed with stage 4 breast cancer. It had already spread pretty widely into my liver, lungs and bones, and later into my brain. And I spent the first 18 months on a series of different drugs, chemotherapies, some of which worked, some of which didn't. Over 3.5 years ago, I was basically given months to live. All the treatments had failed, and then I found myself on a clinical trial here at the Wolfson in Glasgow, and I've been on that trial ever since and doing really, really well. I've been coming here for over 3.5 years. I come every 3 weeks to get my treatment, which involves swapping over my old pills for a new set of pills, which I take daily, and getting a couple of injections and a meeting with a consultant. And it's basically become a bit of a home from home for me. It's such a regular part of my life. And I also come here to get scans every 3 months. So I basically live my life in 3-month chunks of time. was called Timed Out, and the reason it's such an apt title for me is that there's thousands of us patients with metastatic advanced disease, all basically hanging on by our fingertips, waiting for the next drug to come along that might extend their lives. I'm frustrated because it feels like nothing moves very quickly. Trials can take 10 years to reach patients. Nobody seems in much of a sense of urgency about this, so I think we really need to up the pace on it all. So at the TEDx Lab, we covered a range of topics, and we talked about whether the importance of getting patients onto trials. We talked about making molecular testing available to patients, and that's when you take a sample of the tumour, you biopsy the tumour, and you look to see what gene mutations it has, and it's those gene mutations that drive the cancer. And increasingly, there are drugs, targeted drugs like mine, that will target those gene mutations, and that's why I've had such a good response to this trial drug. And we also talked about the importance of having a very good, trusting relationship with your consultant. And in my case, I've been very, very lucky, and my consultant in Edinburgh and in Glasgow, it feels like it's a partnership working with them. They don't make decisions on my behalf. We discuss them and make joint decisions. I suppose for me, the main priorities I'd love to see taken forward are to see if we can create a patient-friendly portal or database of all the trials, academic and commercial, that are available in the UK, so that patients and clinicians can easily find out and offer those trials to their patients. So that's one thing. The second thing is to make molecular testing much more widely available on the NHS. At the moment, you have to pay privately for it, and if we could make that available, I think knowing what treatments might match a person's cancer could avoid the need for people going on to drugs that just don't work for them. And the last thing, I think, is to see if we can encourage a bit more innovation around the drugs that we offer to patients, particularly with advanced diseases. There's many, many drugs out there, but unless they've been through a 10-year clinical trial, all the different phases, patients usually can't access them. So their options when they have advanced disease are very, very restricted. And I just think, wouldn't it be great if we could just, when people are facing the end of their life, be much more innovative and take a few risks and allow them to try some new treatments, because at that point they've probably got nothing to lose and maybe a lot to gain.
Okay, so just a bit of background. As I said, I started as a surgeon, and this is a portrait of Rob Sutherland, who was the director of first the Cancer Research Program in Sydney, the Garvan Institute, and then the Kinghorn Cancer Centre. He was first my PhD supervisor, then my boss, then my mentor, then my good friend, and the godson of my first son, Max. I've been working in precision medicine, precision oncology for over 20 years. We just used different tools, and then genomics came along. We were fortunate enough to lead for pancreatic cancer and sequence 500 pancreatic cancer genomes for the International Cancer Genome Consortium. During that time, I started seeing the results that started coming out. And this is 2009, 2010, and these were patients of mine, patients of my colleagues, patients that we knew. And I thought, well, I really want to know that. So we had to reshape the whole system in order to be able to do that. Not long after that, in 2012, Rob got pancreatic cancer himself. So we did everything we could. We had $100 million US, so we had a lot of money to spend. So we did everything. This is before champions came along with growing xenografts. We made cell lines. We threw lots of drugs at it. We really worked out what we can actually do in precision medicine. And then, having spoken about that a little bit, I got a bit of a reputation around the world for genomic oncology. So the high net worth individuals, the well-connected, the rich and famous would find me, either come over or fly me there to advise them about their healthcare, which is completely boutique, completely off-piste, not part of the health service. Now, that's okay, you learn stuff, but only helps a small proportion of the population. So what we've been doing mostly over the last 4 years is to try and give access to every single individual, whether they be the billionaire, mothers, grandchildren, grandmothers, you might recognize Tessa Jowell there, children, and even different socio-economic classes. Anybody here seen Train Spotting? Yeah, Begbie, King of the Neds. So I think you're right, we should change the name from precision medicine to just proper medicine. Let's get rid of all the names and all the confusion, because this is the way we need to do it. It's a natural evolution of healthcare. What's happened is our ability to measure differences in disease has outstripped our ability to respond to them. These are the questions we all want to know as patients. How do we do this for the individual, not the average? Because there might be a curve like this, and nobody's in the average. So if you think about the questions, how do we use current treatments better? There's no firm data on this, but with the Australian government, more than 50% of the healthcare dollar is spent on the last 9 months of life. That tells me that we're not doing what we should be. Most of it doesn't work. Which cancers kill? We overtreat or we undertreat, or we guess. Pancreatic cancer, they all recur, but we wait for them to recur before we treat them. That doesn't make particular sense. How do they evade treatment? We know they evolve. We start our drug development in nth line, but they've all had different pre-treatments, so they're all completely different. And how do we learn from every patient? Because the numbers we're going to need are staggering. The ability for us to dissect out what these differences are going to be, we saw some examples from Tony, they're going to be staggering. So we need to change what we're doing. I'll come back to tamoxifen, developed in the 1970s. And the reason I know so much about this is Rob was involved in the early development of tamoxifen. And some people might recognize that as a baboon's ass, and it's exactly what it is. And so there was some guy called Brian, apparently, I can't remember his surname, who was a geek, who was in ICI, which became Zeneca, which became AstraZeneca. And he was looking at this tamoxifen. He said, well, it's an anti-estrogen, isn't it? And maybe let's see if we could do any sort of anti-estrogens. And so very unacademically, he just did the one killer experiment, and he suppressed the perineal flush of a baboon in heat. The drug was made in 1966, and this was later in the 70s, and it had been on the shelf. It had been almost deprioritized. So why could we discover tamoxifen then, and now we've been struggling ever since? Because we use the microscope, a 300-year-old technology, great, but also dependent on the nuance of the pathologist's eye. But now, from a molecular perspective, they're different. So if you do a clinical trial, now 70 to 80% are estrogen receptor positive, you're going to get a signal. So in some other instances, in higher proportion, we can start to get signals and work out how we develop. Some examples, like in lung cancer, if we specifically target a particular abnormality, we can get a signal. Even if you triple the survival in a 5% phenotype subtype, you need 30,000 patients to prove a difference. And in answer to your questions about randomized clinical trials, we need to do randomized clinical trials when it's feasible. But if it doesn't, you can't see a signal in 100 patients, what is the point? So we need to fail early, fail cheaply, get a signal. Early drug development is where it's at the moment, because we have so many drugs, we have combinations of those drugs, in just 2 combinations, differences in doses and schedules. We have over 1,000 cancer indications. So you've already got well over 1 billion opportunities with the drugs that we have. And we know many of them are active compounds, but you get an indication and then you never test it in something else because it's just not worth the return on investment, because your patent life is getting too far down the track, and off it goes, it never gets tested. So what do we need to do? Every patient needs a molecular test. We need to get robust data, standardized data. And then we need to share these data globally so we can compare. And the day when we can look at the patient in front of us, check their molecular profile, take their clinical history, and say, there's been no randomized controlled trial for this. But if we look on this database, 50 patients have matched your profile. 40 of them got all different drugs and didn't respond, but 10 of them got this particular drug. So instead of a guess, we're going to try that drug. And then you accumulate those data in what I'll talk about in a minute in either expanded access programs or real-world therapeutic testing to build the data set that's required. So the first thing we started doing is building an affordable test that can be delivered at point of care by the health service, either in a hub-and-spoke model or even in just a single institution. That's because when we tried to set up our clinical trial platform for pancreatic cancer, the tests really quite poor quality, made on 5-6-year-old technology. Even when they're available, they're hardly ever used. And the model that was created by companies that deliver that is made to make money, not to save lives and not to develop drugs. We know if you test EGFR and you give an anti-EGFR agent to lung cancer, you get a significant survival benefit. Similarly with cetuximab in colorectal cancer. But look how many people are actually tested. Back to your point, if we actually did what we're supposed to do, we'd really do really well. So doubling survival is not, I don't think we can even do better. Lung cancer in the community, only 30% are tested. Colorectal cancer in the community, 20% are tested. Breast cancer, when you're probably not supposed to do a test in some instances apart from what's standard, there's a lot of molecular or genomic testing. If you look at the products out there on the market, only more than half of them are not cancer genes. They're only delivered by 1 or 2. There's only 159 genes in common amongst them, and they're tested differently. So how do you compare apples with oranges? And now, since time has elapsed, we understand it's not just about the simple mutations in genes. There's a whole host of other changes that you need to capture. Otherwise, your drug fails because you selected the wrong patients. So the Glasgow Precision Oncology Laboratory, which I started about 4 years ago, aimed to address this question. Very talented genomics people, Susie Cook, genomics informatics from Sanger, Philip Beer, clinical cancer genomics. My second in charge, David Chang, went to address this question. And then what do we do? We have enough knowledge about cancer genomes to know the cancer genes. We're down to finding things under 5% or 3% incidence. Are we going to develop drugs for those when we can't even do it for 10% incidence? It's unlikely. Sure, there might be some more things about the non-coding regions, but we know what we need to find, but we need to find that with as close to 100% accuracy as you can. Otherwise, if you don't pick what's a cancer gene, you'll start fantasizing about something else being a cancer gene, so you need to pick it. It had to work for all cancers, or nearly all cancers, otherwise you're doing 15 different tests. It had to be affordable, and it had to work in the real world. Small samples, routine samples, FFPE. 10 years ago, I said I've got more chance of flying to Mars than have pathology departments move from formalin fixation and paraffin embedding. And the size of the biopsies are really, really small. So as objectively as possible, and this is just for indication, we identify what are truly cancer genes. Red, what's important for drug development now and for treatment. Orange, what is definitely a cancer gene but isn't an actionable event. 178, we still need to learn a little bit more, but there's good evidence to support them. A few that, well, we're not sure about, and these are hematological, and guess what? More than half of genes that are thought to be cancer genes are not actually cancer genes, and this is because they accumulate in agnostic studies with false discovery rates. Only reported in one study with a massive false discovery rate. So we built what we call the clinical cancer genome, or the Glasgow Cancer Test. What we need from the health service: pared down, affordable, deliverable. What pharma needs: discovery or focused discovery for research. And you can imagine a situation where every patient gets a test, pharma recruits them from clinical trials and pays for this test, so the test becomes free. And it's ready to go. Needs some validation in internal labs, etc. But it works on all solid cancers except sarcoma. Works on it, the smallest biopsies, we're almost down to what is the lowest amount you could ever use, which is 10 nanograms. It's being launched globally by our industry partners, Agilent, who have taken it on, marketed it globally on November 4th. We've gone through the initial pilots in England and Scotland, and Italy's pretty much complete. The NHS can deliver the test, they can analyze the test. The International Cancer Genome is going to adopt the larger scale test, because otherwise we're comparing apples with oranges. Genie has 60,000 patients in its data set. Well, not really. It has one lot of 12,000, a lot of 12,000, and 35 lots of a couple of hundred. We need to enable some of the analytics, which is the bigger hurdle, which has not been ignored, and we're moving that to cloud. And we're also having discussions with China, Japan, Korea, and Germany. And why is this important? We know this. Patient benefits are clear. Health economic benefits. We can generate revenue in the health service. Instead of paying the CRO, what does the health service retain? Instead of the pharma company paying the CRO $100,000 per patient cost on average, how can we recoup some of that back and get that to pay for the patients? Especially if they get a complication from the trial, then we bear the cost of that in any case. And we want to build comparable data sets, otherwise we're comparing apples with oranges, because our current model doesn't work. Our pilot demonstrated that we find twice as much as what is currently found, just because of the way the test is designed. And the big question, what's the cost? Definitely cheaper than your phone, Tony. $200 for reagent costs, and some people might say, what are the lab coat costs? But they're already sunk costs. They're already there. You're expending them anyway. In fact, cheaper than just KRAS testing in Italy. And it'll replace some of the old tests and save more costs. One of the interesting things that economic models show a complete decrease in costs, but because it resides in different budgets, we're not going to expend more on genetic testing because it's going to save the drug budget $300 million. Well, that's their problem. We give us the $300 million and then we'll do the test, and that never goes well. We need data. We need to learn from every patient. Do we just start that? We can't. We start with platforms, and we build a platform called Precision Panc, where every patient with pancreatic cancer is offered entry into the platform. And we ask them the first time we see them. That was the biggest innovation, better than all the science and all the publications. Then we don't have to turn the test around in 7 days. I bet we can do the test faster than the NHS can actually get all the diagnostics done. And then we want to move to expanded access programs. Patients want access to new treatments. Drug developers want to develop drugs. These are not pivotal studies. We have thousands, billions of potential combinations. We need to get a high bar signal early on. And so there are many opportunities for expanded access programs, and that encompasses early access, compassionate access, all these sorts of things. Cancer Drugs Fund was a great thing, except nobody collected any data, so generated no value. They just spent money. And then the ultimate, of course, is a self-learning health system, where the health system does it automatically. And I'll just finish with the need for huge numbers. Our goal with the International Cancer Genome Consortium and its new initiative, ARGO, Accelerating Research in Genomic Oncology, is about patients, about clinical data. I get in a bit of trouble when I tell my genome biologists and informaticians, guys, it's not about the genome anymore. And it's not about the computer anymore. It's about the patient. That's why nobody's giving you any money. We want to deliver 1 million patient years of precision oncology knowledge for the world. That's just a start, because we're going to need a lot more than that. We did pretty well in the first 10 years of ICGC. Clinical data, apart from sex and type of cancer, most donors didn't have. We could pick that from the genome anyway, so why did we even bother? From starting for about a year ago, we've got 13 countries involved, 4 in discussions, 24 programs, seeing how we work with 1 million European genomes, other components, and we're at least promised to date at least half, pretty much half of our goals in the first 18 months. But we need to upload those data. So back to those questions. Back to the questions with the scientific slant. And this is what ICGC ARGO aims to address, to amass the data in a standardized way, in a robust way, that builds knowledge, that allows us to interrogate these databases and pioneer and pave the way for us to enact what we want to enact in the future. And thank you very much.